Hello and welcome everyone. In this set of podcasts, we're going to talk about transcription. It's really important we understand transcription at the basic level as we begin to tear apart epigenetics, start looking at the molecular machinery of epigenetics. Because really epigenetics is centered around transcription, or the regulation of transcription. In these next podcasts, we're going to talk about a few main topics. The first is more of an introduction to transcription, or the basics, we'll call it, of transcription. And often when I write transcription, I just write TXN as an abbreviation. So if you see that, you know I'm talking about transcription. Next, I want to talk about initiation. Initiation of transcription, where we'll talk about the promoter in a little bit more detail, pre-initiation, complex, and then initiation itself. Then I'm going to spend just a little bit of time just to kind of bring it back into everybody's memory, talk about RNA modification. And for the most part, what I'm talking about here is modification of the mRNA. And then very briefly, I want to just talk about, or at least provide a short flowchart of how eukaryotic transcription is regulated. And I should also point out, I mentioned the word eukaryotic here, but really what we're talking about in all of this is eukaryotic. We may touch a little bit on bacteria as we go throughout the semester, but when we think about epigenetics, most of what we're going to talk about is in eukaryotes. It certainly does happen to some extent in bacteria and archaea, but we'll focus on eukaryotes. All right, so let's talk about the basics of transcription. And I want to address this question of specificity. Why is there a need because remember from when you took genetics that replication, when the DNA is replication during the S phase of the cell cycle, there's no specificity. It's all replicated. So why not just have the whole genome transcribed? That way you wouldn't have to be specific. Well, off the top of my head, I can think of these two reasons why specificity is needed. Most of our genome doesn't code for proteins. A good part of it doesn't code for anything, doesn't even code for RNA of the non-messenger RNA type. So because of that, transcribing the whole genome would be inefficient. To transcribe all of those billions of base pairs when you only needed maybe 1% of it transcribed would be extremely inefficient and a waste of energy. Another reason, particularly when I'm thinking of organisms or species like humans or any other multicellular organism, different tissues only need some genes transcribed. So if we're looking at the pancreas, there's only a need to turn on pancreas genes and general housekeeping genes, like say glycolysis gene. But there's really no need at all to turn on genes necessary for brain function, cardiac function, skeletal muscle function. There's a need for specificity so that the right genes are turned on at the right time. And related to this, and since I don't have room here, I'm just going to kind of include it with multicellularity, is that below we talked about tissue specific, but also temporal. Meaning that as an embryo, there are genes that need to be turned on that should not be turned on as an adult. At different stages of your life, as you develop from, say, an embryo all the way through an adult, there are just different genes that are necessary to be on that shouldn't be on at other times of your life. So the need for specificity is an important need, and that is why transcription has to be specific. Okay, let's go ahead and get started with the description of the gene. And I'm going to draw two pieces of DNA here where our gene resides. We always write the DNA shown here as 5' to 3' on the top strand and 3' to 5' on the bottom strand. We do this out of convention, but it makes understanding the next process, which we'll talk about later, where we actually make the RNA a little more straightforward. It's important to remember, just to remind you anyhow, that DNA is always anti-parallel, so it's 5' to 3' on one end and 5' to 3' in the other direction on the bottom strand. I want to draw some important structures of this gene. This first part up here, I'm going to write ORF for open reading frame. This is the coding portion of the gene, the part of the gene that will ultimately make a protein. However, as we said previously, we just don't transcribe the whole genome. There's got to be a way to specifically transcribe just this gene. And so it has to be regulated. And we can think of regulation in a couple different ways. The first part here I'm going to write as proximal. Proximal because it's close to the gene. This is what we traditionally call the promoter. This is where we have that consensus sequence called the Tata box. This is where general transcription factors, which we'll talk about later, bind to and recruit RNA polymerase, which is the enzyme that will transcribe this gene. However, we need a little bit more control to be very precise about which genes get turned on and to which extent they get turned on. And so we need more regulatory sequences. And these, for the purposes of our discussion today, come in two different flavors. And they can occur in two different spots. Actually, they can occur in many spots. The key is, though, that they are distal to the gene, or I should say rather, distal to the open reading frame. And they can be either enhancers, sequences, or they can be silencer sequences. They will bind certain regulatory proteins that either help turn on the gene or turn off the gene. Enhancer sequences turn on genes, silencer sequences turn off genes. And remember, these can be at a fair distance from the actual ORF itself. They can be 300 base pairs or even more base pairs away. And though I drew it in a fairly simple manner, this promoter region includes the Tata box and also some additional sequences upstream of the Tata box. But we consider this all to be the promoter and all to be proximal to the open reading frame. Okay, a couple other things I want to point out to you in this gene, and that is, at least one more thing, is this terminator. Remember, we said it's really important to specifically turn off this gene, but we don't want the RNA polymerase to continue to make RNA all the way down forever, because that would be inefficient. Then we'd be turning on genes way down in this direction, and there's no reason to think that those genes need to be turned on just because this gene's turned on. So this puts a stop to it. The RNA polymerase will be coming down this way. When it hits the terminator sequence, there are certain other enzymes involved that will shove that RNA polymerase off of the DNA. It's important to specifically turn it on, but it is also important to specifically turn it off, to stop transcribing at the appropriate time. And while we have this picture up here, I want to go ahead and point out a couple other regions here. I'm just going to show it with like dots here. And that is, we have exons and introns. Introns I signify in between these two dotted lines, and then the exons are here. And I'll make a note down here, introns don't code for the protein. Whereas exons do code for proteins. We'll come back to those in just a moment. Okay, now let's talk about some of the proteins involved in turning this gene on. And we'll go into a little bit more detail in the next section of these podcasts, but I want to introduce some terms to you now. One of these are called transcription factors. Transcription factors will bind to the proximal promoter. I'm just going to put a purple dot here so you can line up with this the purple color of these words. So these transcription factors bind to the proximal promoter to help recruit RNA polymerase. The enzyme known to read the open reading frame and synthesize the messenger RNA. And I'm going to kind of draw this as a big circle here. It binds to these transcription factors, it attaches to the DNA, and begins to transcribe the DNA into RNA. Now, there's a lot more to it, and we'll talk a little bit more about it, but we won't go into elaborate detail about all of that, because really, for the purpose of epigenetics, the main thing is whether or not we ever attach these transcription factors and allow RNA polymerase to bind. Epigenetics will prevent this from happening or allow it to happen. And once this happens, the rest just happens beyond the realm of epigenetic control. We can think of the stages of transcription, and there are 3. Their names are not incredibly imaginative, but they are quite descriptive. The first is initiation, and this is where we're going to spend most of our time, really all of our time. The next stage is elongation, and the last one is termination. Initiation is the process of picking which gene to turn on and recruiting the RNA polymerase to that gene to turn it on. It is a critical part of transcription because it gets the whole process moving. Elongation, as the name implies, this is after RNA binds to it, RNA polymerase binds to it and begins to transcribe the RNA. So elongation is just that steady movement of the RNA polymerase across the open reading frame and the continuous synthesis of the RNA molecule. Termination, as the name implies, is as that RNA polymerase reaches the terminator. Here, it stops transcribing and terminates the process of transcription. The result of transcription is the messenger RNA, and it's going to be the same size as the open reading frame. It will be anti-parallel to the sequence it read, and it always reads the 3' to the 5' sequence going in this direction. So it's going to be anti-parallel, so it'll be 5', 3'. It will have the exon, the intron, the exon, the intron, and the exon. Now, most genes have, particularly in more derived eukaryotes, will have a much more extensive amounts of exons and introns. I just drew this for simplicity with only 2 introns. We're going to come back to this molecule later and talk about how it's processed to the point of becoming the mature RNA that can be translated into cytoplasm. And though I'm not saying it every time, when I talk about, or at least to this point of what I've been talking about with transcription, I'm really talking about the messenger RNA. You can assume for this podcast and this set of podcasts on transcription, what I'm talking about is messenger RNA. the other RNA molecules are transcribed, and in a similar manner, but not identical. Okay, before we move on to talk more specifically about initiation, I want to just briefly talk about RNA polymerases. There are three main types of RNA polymerases. 1, 2, 3. There are also two others that I'll talk about in a moment as well. All eukaryotes have these first three. RNA polymerase is important in making ribosomal RNA. RNA polymerase 2 is important in making messenger RNA. And RNA polymerase 3 is important in making tRNA. The way I've always remembered this, and it sounds a little lame, but this is how I've always done it. The letter R has one hump, so RNA polymerase 1 makes rRNA. The letter M has two humps, shown there, and so RNA polymerase 2 makes mRNA. RNA polymerase 3 makes tRNA, T for tRNA, and the number 3, if you spell it out, begins with T. So that's how I always remember those three. Now, RNA polymerase 4 and 5 are both found in plants. They're not found in other eukaryotes, but they're very specific to plants. RNA polymerase 4 encodes a small interfering RNA. That is very important in protecting plants against viral infections. These small interfering RNAs play an important role in degrading the RNA from these viruses so that they cannot set up shop, so to speak, in the plants. So they're very important in protecting the plants. And RNA polymerase 5 assists RNA polymerase 4 in this process. I didn't mention it, but RNA polymerase 1, 2, and 3 have also other functions in producing the other RNA types that we talked about on a previous podcast. This snRNA, the microRNA, the pRNA. For what you need to know would be just what I have listed here.